Martha Carlin, welcome back to the Keto Camp podcast.
Thanks so much, Ben. I'm really excited to be back with you.
So for those watching on YouTube, you can see that me and Martha both have blue light blocking glasses. So we're going to be so focused and so alert because we're filtering out all the junk light. We look super cool. Martha is not only a dear friend of mine, but also somebody who I've learned from in regards to the gut microbiome, LPS, which we're going to talk about today, Alzheimer's, neurological disorders. And if you didn't listen to the previous episode, Martha Carlin was on the Keto Camp podcast. That was episode 357, which was a little over a year ago. It was an incredible interview. Martha shared her story of her husband, John, dealing with Parkinson's and how she became this citizen scientist and became so obsessed with understanding neurological disorders. And John is doing just great. And Martha, by the way, if you watched that interview last time with Martha, which was about a little over a year ago, and you watch her today, Martha, you have completely changed your physical and mental appearance. You look so incredibly healthy. And I know keto has been a big part of that. You're in my Keto Camp Academy. So let's start right there. How has keto worked for you this past year?
Sure. Well, I mean, it's interesting. I was on the podcast at the end of December 2021, and I had just gotten over COVID. I had some excess weight, I think. I didn't look healthy. I didn't feel healthy. And, after meeting you and really looking more into your approach to keto and how that might help me, I did your first 7-day, let's give it a test. And I was like, I think I want to do this. And it was just incredibly transformative for me because, you've got the support system there, like all the keys for how to do it, the interaction with other people who are, going through the same thing. And, it was just amazing. I lost, I think, 20 pounds. I probably stabilized at about 15. Because I was maybe fasting a little too much early on.
For sure you were.
And getting a little bit too thin, but it just really made such a huge difference in my energy level and how I feel and definitely how I look. Of course, I cut my hair too.
Yeah. And your daughter got married too, right? You went to a wedding last year, her wedding, and you looked incredible. And the dress you wore, and it's just super cool to see that. And it's so important because in the last 12 months, your company and the research you're doing, and we'll get into all of that, has accelerated. So you need the energy for all that has come your way. So what great timing for you, Martha.
Thanks, Ben. And thanks for all your help. It's been great.
Of course. No, I love it. And I've learned we are blessed to have you in our Keto Camp Academy, because when you come on those group coaching calls that we have, you're always sharing with us. I'm always learning. All the students are learning. So it's definitely a win-win, and we're both blessed to have each other in our lives. So let's talk about some cool things. You are always sending me studies here and there. I think this would be interesting for you to read. I think and I love it because you're kind of doing the legwork and letting me review the things that you find fascinating. And you sent me 3 or 4 studies in the last couple of weeks getting ready for this podcast. And we're going to get to that. Really fascinating studies, but we're going to lay some groundwork first and foremost. We're going to talk a little bit about the Krebs cycle. We're going to talk a little bit about how the digestive system works. We're going to talk about LPS, what exactly that is. So where in this discussion do you want to start? Do you want to start with the Krebs cycle? Do you want to start with a little bit of how the digestive system works? I'll lead that to you right now, Martha.
Well, I think, maybe we just start with the digestive system and what's going on in there. We often look at, in disease, we're looking at human cells and what human cells are doing. But in the process of digesting our food, I mean, we really are what we eat. So, that has a huge impact on what's going on in the digestive tract, how that lining is able to protect us from any sort of pathogens and things that are going through the pipe, if you will. And
how thick is that lining?
It's like 1 cell thick. So it's a very, very thin layer of protection that we have that is separating us from the toxins, the bacteria, fungi, viruses, all of that. And, small molecules should go across, but you don't want bigger molecules going across that are inflammatory. So, so that's, people hear a lot about leaky gut. Probably 5 years ago, people thought, like, what the heck are they talking about? Now, there's much greater understanding about leaky gut and what that means for inflammatory inflammagens that are crossing that thin little barrier and getting into the bloodstream and in other parts of our body, where that be joints for arthritis or the brain, and how that's impacting our overall health. So
we have this thin layer, one cell length is just one cell, and leaky gut, I would estimate, and I want to hear your thoughts on this. I would estimate, because it takes years for leaky gut to be actually diagnosed, if it ever is. I would estimate that 80% plus of the American population has some variation of leaky gut. Would you agree? Would you say it's less? Would you say it's worse?
I'd say that's probably a pretty close estimate when you look at the disease burden we have and just what's been happening over the last 20 years to our food supply. There are so many things in our food supply that impact that permeability that it's, it's very hard to stay away from all the different things that can have an impact.
So true. It's I'm always astonished, Martha, with all the hits that we take as a human body, how we just still kind of push through. Human body is so strong. The way we were built, God has built us so incredibly strong. We take so many hits and we still move forward. So with leaky gut, we have a scenario where these larger particles, like you mentioned, they go undigested and they go through this mucosal barrier, because there are holes in it. That's kind of what leaky gut is. So these larger particles now enter the bloodstream. What happens next? Why is this problematic?
Well, so the one in particular that we've been looking very closely at is called lipopolysaccharide, LPS for short. And that is actually a cell wall component of gram-negative bacteria. So as those bacteria break down and die, fragments of their cell wall are left, and this LPS can cross over and get into the bloodstream and cause inflammation, depending on where it goes to. And, what was so interesting about learning about the microbiome and seeing gram-negative bacteria as one of the potential indicators in Parkinson's, and then seeing that in a number of other diseases, I actually found a researcher in Israel who was a dentist. He just died last year in his 90s and was still publishing papers all the way up to the end. And one of his areas of expertise, his name is Isaac Ginsberg, and he had this comprehensive theory around, he worked a lot on sepsis and infection, but that after you have an infection, this bacterial flotsam and jetsam, or viruses or bacteria or fungi, the components are still circulating in the system, causing these inflammatory cascades. And, if you don't get the healing part of the cycle, so there's two different kinds of macrophages, M1 or the killers that try to take care of something, and then M2 comes in and does the healing. And if you don't get that healing process going, you have this chronic inflammatory state that just continues to build over time. So you don't have acute sepsis, but you have this low grade kind of septic state. And what happens in the body when you get an LPS challenge is it actually affects the macrophages in a way that there's something called LPS tolerance. And LPS tolerance is kind of like a paralysis of the process of eating up those macrophages and getting rid of the debris, because the macrophages are like your Pac-Man. So they're coming in, eating stuff up, and then there's a whole process of degrading that. And if that process of degrading that gets slowed down or stopped, I would sort of equate it to maybe like a traffic jam. Like you've got all these Pac-Man, they're eating stuff up, and somebody puts up a stop sign or a yield sign that slows that process down. And so you start getting this inflammation that's this chronic low-grade LPS.
That's the worst kind. So how do you test for this to know if you have high levels of LPS, number one? And then number two, what are the leading causes to why LPS would be elevated? So first, how do you test, and what are the leading causes to it?
So you test for it. There's a blood test called serum LPS. It's not something that is done very frequently. We did it as a part of a clinical trial that we did. I'm sure you could get it at one of the testing companies.
LabCorp request, probably. Yep.
I'm sure that they would have it. And they would do something to that effect, most likely if you had a acute infection in the hospital, to test for, sepsis or something like that. But you get it.
That's the only way.
Yeah. Okay.
That's the only way to test it. Get a blood test of serum LPS. There's no other way.
Well, I don't know if there is another way. That's the way I know. I haven't looked for another way, so I'm happy to look for another way and send you a note if I can find it.
Yeah, I don't know if there is another way, but that's an easy way. It's just a blood test, a blood draw. So that's how you get that done. So what are the main reasons why somebody might have high LPS levels?
Well, it's the gram-negative bacteria. So and I hope you're not going to ask me the names of all the gram-negative bacteria, because I didn't memorize them here. But
I don't expect that.
But what's also kind of interesting in this, back to the macrophages and not just the LPS. So LPS is part of that, but there are other proteins or peptides that these different bacteria are producing that can also trigger via something called molecular mimicry. So they'll create an antigen because your body recognizes, okay, this is a protein from a pathogen, and it will create an immune response and an antibody to that sequence. But a lot of those sequences in bacteria are very similar. The term is homologous. They're very similar to human proteins. And so if you get a buildup of those, then you can also end up with various autoimmune diseases. And so that's another area that we've been looking at, is from our microbiome data that we looked at in people with Parkinson's, different of these heat shock proteins that create an antigen, one being something called cardiolipin. The bacteria have a cardiolipin in that membrane also. And so the body can create a reaction to cardiolipin, which is actually the mitochondrial membrane. So all these little, pieces of flotsam and jetsam, as I call it, can create mitochondrial stress.
Interesting. Yeah. And there's a study we're going to get into all about that. And cardiolipin is very important. It's this lipid raft, and it's really important for the integrity of that mitochondrial membrane. I remember I was interviewing a gentleman named Dr. Now I'm forgetting his last name. We were talking about fish oil. This is several years ago. And we were getting into some of the research on the dangers of too much fish oil. A lot of people are just taking way too much. And he had referenced a study showing that when you overdose on fish oil, omega-3, it actually pushes out the cardiolipin and creates a very, very too fluid mitochondrial membrane, creating a different problem than most people hear about, because it's more of an omega-3 dominant, and we hear more about like omega-6 dominance. But either way, the cardiolipin is very important for the integrity of that mitochondrial membrane. So let's get into some of the studies. Mitohormesis, is that the one you want to start with?
Sure, we can start with that.
Okay. So you sent that study to me about how mitohormesis reprograms the macrophage metabolism to enforce tolerance. What did you see in that study? I want to take a quick break from the video you're watching to share something with you. If you've been following me on social media or any of my online platforms, you always see me wearing these glasses. And I always get asked the question, hey, why are you wearing those tinted glasses? Well, these are blue light blocking glasses, and they are very beneficial to protecting brain function and being productive throughout your day. If you're like me, you are surrounded by artificial light every single day, from the computer screen, phone screen, artificial lights, and your brain needs to filter out the junk light. We're just not designed to filter that out, and it's a lot to filter out. So the analogy is this: if you had a computer open in a web browser with 100 tabs open in your web browser, and hey, that's probably you, your computer is going to function slow. It's going to dysfunction, and it's just not going to be optimized. But what happens if you take those 100 tabs and close them all out and just maybe leave a couple tabs open? Your computer is going to function better. This is the same thing with your brain. When you use high quality blue light blocking glasses, you filter out the extra junk light so your brain doesn't have to do so. A result, you feel better, you think clear, and you have a more productive day. And that is why I wear these glasses. The glasses that I'm wearing and that I recommend is from Bon Charge. They are a sponsor of this channel, and they make some really cool, high quality, well-researched blue light blocking glasses for the daytime, like the ones you see me wearing now, and for the nighttime that are more orange, red tinted. So at night, I switch over to those. They have cool styles like the ones you see here, and also clear frames as well. They have reading glasses. You could get your prescriptions in those glasses, and they have a whole range for you. So all you need to do to get your hands on a pair of Bon Charge glasses or any of their wonderful products is head over to boncharge.com/KetoCamp. Use the coupon code KetoCamp at checkout and save 15% off your entire order. We'll drop a link and the coupon code for you down below in the video notes. So go ahead and get yourself charged up with their awesome products. Okay, let's go back to this video.
So, I mentioned a few minutes ago that when you get an LPS challenge, you can become tolerant. You get LPS tolerance because the body is designed in a way, like usually when we get an infection, like if you get sepsis, it's actually your immune system's response that kills you. It's not the pathogen itself. It's, drowning in your own fluids that are trying to deal with the pathogen. So, there's this, I guess maybe it's a valve release or something that in terms of a good parallel that says, okay, well, we don't want too much inflammation. So let's set the system up to tolerate a certain level of inflammation. Mm-hmm. And so you get this LPS tolerance. But if you think about it, then when you have LPS tolerance and it paralyzes the macrophages, you're getting this traffic jam. So over time, the more and more traffic that builds up slowly with that LPS, the more problems you start to see, which is why if you think about what's going on with the American diet, we don't recognize it immediately. It's this sort of gradual thing that happens. Well, this, mitohormesis, and I know you talk a lot in Keto Camp and talks that you do, and, Dr. Mindy talks about the importance of hormesis and intermittent stress for challenging the mitochondria and, making them flexible. And so this process of mitohormesis can reprogram macrophages, but LPS can also reprogram macrophages. So you can get this sort of, I'll say, a wobble back and forth, particularly if you have this constant buildup of LPS. So you've got this constant inflammation and you want to dampen that down, but you also want to get rid of the traffic.
Mm-hmm. Yeah, we don't want a traffic jam. That doesn't sound good. So that's, if you don't take care of it, that's where we're seeing it lead to things like neurological disorders, and even there's a connection to diabetes and other conditions, correct?
Correct.
Well, that's not good. So, okay, traffic jam. So it's kind of twofold, right? So what do you want to get into the solutions here, or do you want to talk a little bit more about the problems in the other studies?
Well,
Where do you want to go?
I want to talk about something interesting. A new book that's out. I'm gonna hold it up and plug this.
Transformer by Nick Lane.
By Nick Lane, and that's about the Krebs cycle, which we were going to mention earlier. So the Krebs cycle is actually how energy gets produced. It's a circle of biochemistry, and in that process, a lot of different metabolites are used and produced and sort of go off in one direction or another. But it turns out that bacteria can run the Krebs cycle backwards. So
fascinating.
This was an area I started looking at probably, 6 years ago, when I was trying to figure out what was going on with energy in people with Parkinson's, because they just sort of bonk sometimes. Same thing with people with chronic fatigue. And if you think about like a cycle that's going in a circle in one direction, producing energy or producing the metabolites that are used to produce energy, and then you have a cycle going in reverse, you're using up, and there's basically something called a feudal cycle, where you're feeding these intermediates back and forth to each other without going on to produce the energy that you need.
Interesting.
So yeah, his book was, I love his books, but it was really interesting to me too, because he was looking at the difference between nuclear DNA and mitochondrial DNA, and I know we talk about mitochondria a lot. Well, they basically took a set of flies and they all had the same nuclear DNA, but they had different knockouts in changes in the mitochondrial DNA. And they were doing this study where they were giving the flies NAC, and this one set of the female flies all died when they got NAC, because there was this mechanism in the process where these signals cause a repression of respiratory enzymes. So it starts dampening down respiration, and basically they sort of suffocated from the inside out. Because respiration wasn't working properly. Well, that, then that tied back over to this other stuff I had been looking at, where in bacteria, I was looking at, lots of studies have been done on bacteria and what their preferred substrate that they grow on, different types of sugar and stuff. And I had found that glucose repressed respiratory enzymes. So as long as there was glucose present, the respiratory enzymes were repressed. So, that sort of played in with that fly thing, is like,
similar to the NAC on those female flies.
Right. Are we somehow, through this high sugar diet that we're eating, are we repressing our own internal respiratory enzymes in the mitochondria? And then that, of course, tied back to the work of Richard Johnson, which you've had on your podcast before. Nature Wants Us to Be Fat. And the epiphany I had reading his book, which, and I talk to people who are doing keto and all these, they're like, I'm still having problems. And I didn't realize we produce fructose inside our body when we're under stress. And so you could not be eating any fructose, but if you're under a lot of stress, and that can be mental stress, that can be emotional stress, that can chemical, physical, nutritional, all these kinds of stress can then create this endogenous production of fructose, which then alters, one of the key metals in the body, copper. It chelates all the copper.
Okay, a lot to unpack here. So, it's interesting that you have found that glucose acts very similar to what the NAC did with that study in terms of like repressed respiratory system, essentially lowering the energy production, right? So it makes sense though, because when you do keto, when you're in ketosis, and we know ketones are these signaling molecules, they're actually communicating with the mitochondria, but it's also like mitohormesis too. It's actually, ketosis is kind of like a mitohormesis. It is actually, where it's a stress to the mitochondria, which forces them to get stronger, right? So we know that stress is only bad if you don't adapt to it. But when your body adapts to it, mitochondria get more robust. But just looking simply at the electron transport chain, when somebody is on a high sugar diet, a standard American diet, and they're utilizing glucose primarily, they're producing about 32 to 36 ATP, right?
Right.
And then when you compare that to somebody who's in ketosis, it's 400% more. It's 132 plus ATP. So I'm guessing, based off of what you just shared, part of that reason why you're producing less energy with sugar is because of this repression of that respiratory system.
Yes, and all those enzymes in the respiratory chain are copper dependent. So when you start
Ah,
running on iron, and I think Morley might have discussed this with you, I think that ATP
you did.
level
Yeah.
can drop down to, instead of 32, it drops down to, is it 2 or 4? It drops down way down.
It drops down to single digits, because it chelates the copper, which interferes with the energy production. Wow, yeah, that makes a lot of sense. So, okay, but then we have. Let me stop for a second before I'm going to say that. So, fructose. Does that mean we should avoid fruit, or does that mean fruit in excess is really the problem, but fruit from time to time should be totally fine?
Well, I mean, I'm not the guru of this, but I think if we look at how we lived historically compared to how we live in the last 50 years with refrigerators and airplanes moving food around, um, we can have for any fruit we want anytime at the grocery store. And
True.
if you think about when fruit ripens, it ripens at the end of the season when you're normally going into a period of time where you might be under food stress historically. So, maybe at that point in time, that's when you need to eat some fruit because it's local, available, fresh, hasn't been shipped from all over the world. And that's the signal to your body of, we're going into a different time and you should switch to a different kind of cycle. I also I talk a lot about food just being information. I mean, we're made of genes, food's made of genes, bacteria are made of genes, and it's all flowing through this pipe. And, it's not as there's a straight fine line between, all of those pieces of genetic material.
Yeah, so many moving parts, which is why, just telling people to eat less and move more doesn't help. It just doesn't give you the full picture. Not denying calories. I'm just saying it's not important to just track your calories. There's so many moving parts, and we haven't even yet scratched the surface, meaning like science on this incredible human body. We're just still figuring things out. Okay, so I agree. I think fruit could be great, and I have from time to time. But if you think about what you just said, we're in this day and age, we're eating fruit winter, summer, spring, fall, year round, anytime. You can get bananas, you can get whatever you want. And then on top of that, Martha, we know it's not just people who are eating the whole fruit, they're juicing it and they're getting this high glucose spike. So that's not what we want to do. We want to have fruit when it's whole, have them seasonally or time to time if you have a healthy metabolism. So that's kind of the goal, to build up that metabolism to introduce fruit, which is what I do personally. So,
Well,
continue.
yeah, I'll just add to that some of the pectins. So citrus pectin and apple pectin,
Yeah,
they can also be important components to membranes or helping with membrane assembly. So, it is something you want to, it's not like you want to say, I don't ever want to have fruit.
correct. So, what are your thoughts on Paul Saladino? 200 to 300 grams of fruit every day, and meat, fruit and meat.
Well, I think there's just everything in moderation, shall we say. Like that's
Yeah.
just probably the best advice you can give somebody. And that's also why I really love the Keto Flex program, because it is, we're doing things that have, a cycle and a balance back
Yeah.
and forth, and we're not overdoing any one thing. Because you can swing the pendulum lots of ways, and that's a lot of fructose.
It is. And Paul is, Paul's great. He's super smart. I like him a lot, but he's also very active, metabolically healthy and flexible. Somebody, 88% of the country is not healthy. Their metabolism is just, it's challenged right now. And they could do better doing a little bit more keto fasting, and then eventually start flexing, which is what we teach. And for example, like January just ended, right? And I just, with a bunch of the students, I don't think you did it this time, but we did carnivore. I'm actually still going on day 33, and that's a good time to do it. The winter is typically when our ancestors did more animal based, right?
I did it for about 10 days, but then I was traveling a lot, and it's just really hard to do when you're staying with other people.
It's restrictive. Yeah. What's the longest? I think you did it for a full month with us last year, right?
I did do it for a full month last year, but I did the phase 4. Like I've never done just like the level 1.
Yeah.
Where it's
I mean, neither. I do like
elements.
a variation. So you did level 4 where it had, mostly meat, but some like plant-based sauces and spices. That's still great. Yeah.
Although it's the
Okay.
perfect time
You
to do it, because we have cows on our farm that we've grown ourselves on the grass.
That's
And
awesome.
we just got one of our cows back. So.
That's awesome. I know where I'm going. If there's a civil war in the US, I'm headed to Martha's. Okay. Speaking of LPS and A1C and high glucose, you did a fascinating study that was conducted in Cuba, I believe, and you looked at A1C, you looked at LPS, and you looked at a few other things. Could you talk about that clinical study you did? Hey, I want to just interrupt the video you're watching real quick to share something with you. As somebody who's been in the keto space for 14 years, I've taken thousands and thousands of people through a keto protocol. I've seen the number one problem and struggle people have on keto being this electrolyte dumping process. Allow me to explain real quick. When you lower your carbohydrates and lower insulin, you're going to release excess water weight, which is terrific because you're going to look lighter, less puffy, and feel lighter. The problem is this: the kidneys also release excess electrolytes, and you go through this sort of diuresis process leading to symptoms on keto. And what is the solution? Keeping your electrolytes up. And when I talk about electrolytes, there are good ones out there. Then there are optimal, incredible products. The one that I use is from Upgraded Charge. One of the reasons why I love this product is that it has a unique proprietary blend of nanoparticles that have the ability to get into your cells, which is for maximum absorption. It contains magnesium, zinc, sodium, and potassium, and it tastes pretty good. Kind of tastes like a non-alcoholic margarita, and the flavor comes from the lemon peel. Simply use this by adding it to your water or sparkling water and get your electrolytes up. Replenish them. This is key to feeling incredible on keto. Upgraded Formulas has hooked you all up with a sweet deal to get their Upgraded Charge or any of their amazing products at 15% off. All you need to do is head over to upgradedformulas.com and use the coupon code KETOCAMP at checkout and get 15% off your entire order. I will drop a link for you with the coupon code in the notes down below. All right, let's get back to this video.
Sure. So we took our Sugar Shift formula. I'm not holding it up, but the Sugar Shift probiotic formula, which was the formula I originally made for my husband. And we found that it was helping people get into ketosis. It was helping people improve their blood sugar and all that. So we decided to do a clinical trial in a diabetes cohort. So we had 30, so it was a double-blind placebo-controlled trial. So the placebo was actually all the ingredients in the formula except for the bacteria. So it was the same mix except for the bacteria.
Got it.
We could see that it was the bacteria making the difference. And we did that for 90 days, taking 2 capsules, one in the morning and one in the evening. and measuring. So they measured fasting blood glucose, postprandial blood glucose, and serum LPS, and a measurement that's not used here in the United States, but it's called HOMA-IR, which is a measurement of insulin resistance.
Mm-hmm.
And we did microbiome data. So before and after microbiome and
Stool test.
Stool test, yes.
Awesome. Okay, and A1C, I think, right?
And A1C.
Yep.
And so, for anyone who's not familiar with A1C, although probably most of your people are, that's, the sugar molecules that are on those cells is what they're measuring. And so, the glycosylated molecule. And so those blood cells, your blood cells typically will last about 120 days, although it varies from person to person. And we only did our study for 90 days. So what we found was the HbA1c stabilized in the group that was taking our product and continued to rise in the group that was not taking the product. But then at the end of the study, since we determined, 90 days with 120-day blood cell life probably wasn't long enough, we unblinded the study and kept 10 people on the product for another 10 days. I mean, not 10 days, another 90 days. So 180 days total for that group of 10. And what we found at the 180-day measurement point was there was about a 14% drop in HbA1c. Wow. But at both 90-day and 180-day marks, there was a marked drop in the serum lipopolysaccharide. So we were significantly, reducing that burden of gram-negative bacteria lipopolysaccharide. So
fascinating. So all they did was take one capsule of Sugar Shift in the morning, one capsule in the evening, and those were the results. That was the only change they made.
So the cohort was split into, so of those 30 people, some were on insulin, some were on metformin, and some were just on diet. So, but that was the only, they already had those interventions in place, and there was no other instruction of, change this or do this. And, in Cuba, they don't have as much access to, say, protein or something to change to a keto diet. So it is a population that's generally on a higher carbohydrate diet already. Although I would say anybody who's participating in a study is probably going to be slightly motivated to do a little more moving, just because, they do.
That's fair. Totally. How many, I think I missed it. How many total people were in the study?
So there were 30 in each arm. So 30 on product and 30 on placebo.
So what is it in Sugar Shift that helped with the LPS, helped with all these other markers? What were the specific bacteria that helped with this?
Well, so what we found was a decrease in some of the inflammatory bacteria. So Akkermansia, which sometimes is shown as a marker of inflammation. We also saw an increase in Faecalibacteria and Roseburia, Roseburia is a carbohydrate degrader, but has been shown to be a beneficial microbe in the gut. And then the biggie was an increase in Bifidobacteria. So Bifidobacteria is I call it the baby bacteria, really. If you're breastfed, there's a single, which, we don't have as much of here in the United States anymore, but in indigenous populations, breastfed babies will carry a specific Bifido infantis strain that is a single colony that's like training the immune system and doing all this incredible stuff. There's actually a company, if you have anybody who has a new baby, they have it's called Evivo, and it came out of UC Davis for that particular strain of Bifidobacteria. But Bifido has been shown to be protective in a number of different diseases. There's a strain that Morinaga milk in Japan has looked at with improving mild cognitive impairment. And they've also shown in the last 2 years in a lot of the microbiome research around COVID that people with low bifidobacteria had a higher or worse outcomes or more problematic COVID. And in Parkinson's, people have low bifidobacteria, which is interesting because we looked at the different species of bifidobacteria. I'm getting off my study here, but in our Parkinson's data, there was one strain of bifidobacteria that they did have that is resistant to glyphosate. All other of the strains of bifido are mostly known to be killed by glyphosate.
Huh. Interesting. Okay. And what about mannitol? Because you were teaching us on the group call a few weeks ago about mannitol and its antioxidant scavenging properties. So I'd love to hear more about that.
Yeah. So the product was actually designed originally to produce mannitol in your gut. So it takes glucose and fructose, and the bacteria transform it into mannitol, and you get butyrate, and which is anti-inflammatory and feeds the lining of the gut, and a number of other anti-inflammatory compounds. But mannitol is a really powerful free radical scavenger, and it also helps with. It's the most abundant sugar in nature, and in plants, you'll find more mannitol in fruits and vegetables that are under osmotic stress. So something like a watermelon or celery will have more of it, or even like a cactus or something like that, where it helps with that draw, the osmotic flow. And it's also interesting that mannitol is used sometimes in research when people are studying the mitochondria. That's one of the media that they can put the mitochondria in to keep the mitochondria happy. So it crosses one of the membranes, but not the other membrane. So, and I had another brain researcher tell me, oh yeah, mitochondria is a really interesting molecule because, so it's used in brain trauma. If you have swelling.
Mannitol.
Yeah.
Mannitol, you mean?
Yeah. Mannitol is used to reduce the swelling on the brain, but he told me it causes a tsunami in the cerebrospinal fluid. So, one of the things I'm looking at is that free radical scavenging maybe going on, like if you're flushing the cerebrospinal fluid. I don't know. So we're still kind of looking at ways that we can do deeper science on the mechanisms when we have the funds to do that. But it's a really interesting, because it behaves differently than a lot of the other polyols. Most people run from the polyols, and the polyol pathway can create a lot of problems in the body. But mannitol is different. And humans, if you take mannitol, most of it will come out in your urine. So it's not absorbed. It's often used as a substitute, a sugar substitute for diabetes.
Sugar substitute.
Yeah.
Do you recommend using that as a sugar substitute or not?
Well, my husband does take it, because our design of the formula was based on this research that showed that mannitol by itself, that sugar alcohol in an animal model, could stop the aggregation of the proteins that were a hallmark in Parkinson's, and then pull them out of the brain of the mouse and get rid of them. Wow. So that's how I came up with the idea. And so then there was a group in Israel that set up something called ClinicCrowd, and they were crowdsourcing data from people with Parkinson's taking mannitol to report if there was an impact on their symptoms. And for a period of time, there was quite a bit of information about it. You could go to the ClinicCrowd website. Now it's very difficult to find. There's a Vimeo story from the I think he was a technology entrepreneur who started this whole process because he had Parkinson's, and they interviewed different people and how remarkable their change in symptoms was from the mannitol.
Wow.
So, again, everything in balance, but John does take about 2 tablespoons of mannitol a day. So,
That's fascinating. Okay, so what about somebody who, somebody who's not at risk for Parkinson's? They don't have Parkinson's. Would you suggest using mannitol as a sugar substitute, or would you go with something like stevia, monk fruit, or maybe erythritol? Or do you think mannitol is totally a good option?
well, I think mannitol has some pretty interesting properties.
Yeah. Well, you're changing my mind because I in the past would not usually it would not be my go-to. I would, lean towards stevia or monk fruit, but you're kind of changing my mind here.
Well, I also had to I was using monk fruit in a number of different things, and my oldest son, who helps me in the business, has a lot of food allergies, and he cannot eat monk fruit, which kind of was like, what is it about monk fruit? Or is it how the monk
Yeah,
fruit is you also have to think about how things are processed.
for sure. They're all processed. That is correct. Yeah. I think we could agree though that these are better options than like sucralose or aspartame.
Yes,
Those are ones we want to stay
definitely
away
agree
from for
on
sure.
that.
What about the role of antibiotics? I want to talk about this a little bit because it's just so common, Martha, for crazy things. Doctors are just so trigger happy to just prescribe an antibiotic, and there's a time and place for them for sure, but not the way we're prescribing them to this day. And how long does it take to for the gut microbiome to recover from one round of antibiotics? And what is it doing to the gut when we're taking these antibiotics on such a consistent basis?
So, there's lots of research on what it's doing, and probably one of the best labs to look at is Dr. Martin Blazer. He was at NYU. Now he's
Yeah,
at Red.
it's got a great book. Yeah.
Great book about it, and has continued to do fantastic research on the impact of antibiotics. And in particular, he showed in some of his studies that macrolide antibiotics, and off the top of my head, I can't think of brand names, but I think maybe a Z-Pak might be a macrolide, that the microbiome never recovers from a macrolide.
Never.
And
He used that word, never or rarely?
he said never. I believe he said never in the
Wow,
book.
that's an absolute.
And that sort of sent me back to our friends, the mitochondria. There's a group at, I think it's the University of Oregon, who did a gene transcription paper on antibiotics and the microbiome, and overlaid, okay, this is what your mitochondrial gene expression looks like. And then when you take antibiotics, this is what happens to the mitochondrial gene expression. I'll send you that paper, Ben. And then,
Please, that's fascinating.
and then
So
here's what happens with the resistome that is left behind, those bacteria that are left behind. What happens to the mitochondrial gene expression? And you actually have more problems with the resistome that's left behind than you have with the antibiotics. But to sort of connect that back to John, we were doing microbiome. I mean, he's had probably 150 microbiome tests, whole genome. I mean, that is thousands of dollars of research that we did on him. And, we had been looking for markers in Parkinson's, but especially looking at him. And, then we showed with him taking Sugar Shift, how it was moving back to a healthier microbiome. But in that process, we also identified a intracellular parasite that was in both his fecal microbiome and his oral microbiome. And one of my advisors is a world-renowned infectious disease doctor. And she said, whose sample is that? I said, that's my husband. She said, well, that could be causing, nearly all his symptoms. And it was a parasite called Toxoplasma gondii, which it turns out about a third of the population is infected with. But
of the U.S. population?
Yeah, I think it's global, but the U.S. too. There's a big study about the impact on neurological disease from the University of Chicago related to toxo. But so while we were measuring all this stuff with Sugar Shift, we actually put him, we went to the doctor, they treated him with Bactrim, which is a antibiotic. It's like two different antibiotics, a sulfa drug and something else. And
Yeah.
it was pretty interesting because his memory improved like within a week. His memory was much sharper, but his microbiome just like dropped and was down to, so many things were gone, but there was one bad bug that it dropped a tiny bit, but not that much. And it's called Porphyromonas. And I know you talk to people about the importance of oral health. Well, Porphyromonas is an oral pathogen. And that's also a marker that we've found as a potential, one of the potential contributors in Parkinson's.
Wow, that's found in cavitations, and it's an anaerobic bacteria found in the gums. Oh, interesting. So the antibiotics that they gave John, your husband, helped short term because it wiped out that parasite. Was it a parasite you said?
Yep.
And his memory came back, but short term benefit, he did some long term damage because it wiped out, his diversity decreased and all this good bacteria decreased. So you learned something from that.
Right. So we had to, rebuild again. And then the December when I had COVID, he had COVID too. And, he didn't fare as well as I did. And he also
I remember.
He had a gram-negative bacteria infection and took an antibiotic then. And it's taken us close to a year to get him back, where he was. So it's, like if you look at, I forget what the theory is in ecology, but there's this theory about what happens in a system when it gets disrupted. You have a disruption and then it settles back, but not exactly to the level where it was. And then you have another disruption and it settles, but you're constantly leveling out to a different new normal. And it never really ever goes back.
It's wild. And if you just think about, oh, you have a UTI, here's a round of antibiotics. Oh, it's not going away, here's another round of antibiotics. Oh, you have whatever silly condition going on.
What people really don't realize is how much antibiotics are in the food supply. They're given to animals as growth promotion. That was a hot topic 4 or 5 years ago. They changed some regulations, but it really hasn't changed much about what's been going on. But, all these great convenience foods that we get, prepackaged chopped vegetables, lettuce, triple washed lettuce is washed with chlorine. You get a tub of hummus. If you didn't make it yourself and you bought it at the grocery store, that's basically a Petri dish. So what do you think they're putting on top of the Petri dish? Anti-enzymatic things so that it won't, grow something on it. But then you're eating anti-enzymatic things, and enzymes are what break down your food. So it's like,
I'm glad you brought that up, because somebody watching or listening is thinking, well, I've never gotten, or it's been years since I've gotten an antibiotic, but the food supply, these animals that are pumped full of them, but also the vegetables. Let me ask you this. You mentioned the lettuce and the hummus. What if it's organic, 100% certified organic, non-GMO? Is it still a problem, or just less of that contaminant in there?
Well, I used to think it was less of that contaminant. It probably is less of the contaminant. But I did a talk for the Natural and Organic Association about 3 years ago about the uptake of antibiotics from manure into roots and shoots of plants. And organic produce is primarily grown with fertilizer that is conventional manure. So it has the antibiotics in the manure. It has the glyphosate from the feed. I've actually been working on a study with Dr. Don Huber, who's probably one of the global experts on glyphosate, looking at an organic farm and what was happening to one of their crops where it couldn't form the proper structure. And that was because calcium, manganese, and copper were chelated from the plant. And that was basically from the conventional manure we traced back to the glyphosate that was in the conventional manure.
Interesting. Okay.
So everything's connected, Ben.
Everything's what?
Everything's connected.
Everything is connected. So there's different levels here. So I don't want to, for those listening and watching, I don't want you to feel like, oh, what's the use if I can't get away. There's different levels, but even if we're getting grass-fed beef and there might be some antibiotics or whatever in the fertilizer, we might be getting some of that. It's a lot less than grain-fed, pumped full of antibiotic, caged animals. So there's levels that are better. Of course, the best level would be to grow your own food and go live on Martha's farm, but I get that. I live in Miami. I'm not going to do that. So there's levels. But here's the great news. There's things you can do with supplementation, diet, nutrition to repopulate and heal the gut. Fasting is terrific for the gut. So, we just want to do our part and do the best you can with the food supply and all that. And then do keto, do fasting. If you could get Martha supplements, which I'll talk about in a second, there's things you can do there. So, I just want to give that hope to the audience. I don't know.
Well, I give hope too, because, we also have, another company that does agricultural products where we are working with farmers with our soil probiotics to remediate hydrocarbons and glyphosate out of the soil. Because, it is all connected and it can be fixed, but we have to take action and make people aware of it and speak up.
Yeah, absolutely. I love, I don't know if you've checked out, Paleo Valley has a sister company called Wild Pastures, and they're actually a sponsor of this. And they actually are like 100% legit. Like their fertilizer is free of antibiotics, no glyphosate. Like that's a right sort of farm that does it the right way. It's regenerative farming. So there are companies that are doing it. It's rare. But to Martha's point, they're all connected. It's all connected, and just keep doing the best you can. So I know we spoke a lot about your product, Sugar Shift. By the way, I need some Sugar Shift. You could get that, Keto Campers, if you're watching on YouTube or listening on the podcast. We'll drop a link down below. But Sugar Shift is the main product we spoke about today. It's the one that you did the clinical studies in Cuba. You could find that product over at biotiquest.com. That is BIOTIQUEST.com. I'll drop a link in the notes. And we have a 15% coupon code that Martha has graced the Keto Campers, and the code is KETOK15. That is KETOK15. We'll put that in the notes down below. But you also have, a probiotic for the immune system. You have a probiotic for the cardiovascular system. You have a probiotic, speaking of which, that I always send people to get if they've gone through a round of antibiotics, called the antibiotic antidote. And that was formulated to repopulate what the antibiotic has gotten rid of, right?
Right.
So you have that. You have one for sleep. Perfect Peace coming soon. What's that?
Well, that's to support mood, GABA production. I'm going to bring that out this year, but we're delaying it just because we've had so much growth on the other products. Okay, like we don't need to put another thing into the mix here until right now. So, but it is to help with mood, and I have a couple of entrepreneurs I know who are working on other products for brain health and mood and depression and that sort of thing. So
that's awesome. I love it. So yeah, you can get any of Martha's products, biotiquest.com. Code is KETOK15. Anywhere else you want to send them? Any other website or any other social media?
Well, if anyone is interested specifically in Parkinson's, I do have a blog called Martha's Quest, where you can, I write about nutrition and exercise and the microbiome and Parkinson's disease. So you can get a lot of resources there, and connections to doctors who are looking at specific, a little more out of the box type things in terms of Parkinson's. So that's a great place for people to go. And if they want to get nerded out on poop collection, my BioCollective website, but we don't really collect poop anymore.
Yeah, we talked about that last time. You collect a lot of poop in the past. So we'll drop links for all that down below. If somebody is interested in learning more about what you know and what you have learned in terms of books, you mentioned Nick Lane's book, but any other books that come to mind that you would just suggest for the audience to explore?
Well, there's a great book from John Cryan and Ted Dinan out of the University College Cork in Ireland. They do some of the best gut-brain research, and it's called The Psychobiotic Revolution. Does a great job of explaining more in layman's terms how the brain and the gut interact and things that you might think about to do there. I think those guys are great. Emran Mayer's book, I think it's the mind gut connection or something to that effect. He's out of
What's the author's name?
Emran Mayer, Mayer. That's a great book. And then
Yep, the Mind Gut Connection, Emran and Mayer, MD. Mind Gut. I'm adding this to my cart right now. It's got a lot of reviews. Wow.
yeah, and then Rodney Dieter, the human superorganism. So he talks a lot about. He's actually I think he was a veterinarian. He's retired from Cornell, and he shows this rise in what they call non-communicable diseases like diabetes, and the connection to what's going on in the immune system and the food supply in our diet. And I've been very lucky a couple of times to actually meet with him or be on a panel or at a conference with him, and he's just
What's
a brilliant,
the book again?
The Human Superorganism.
Okay, got it.
It's a bit older book, but I mean, it really.
Yeah, you're inspiring me to you are inspiring me to do like a whole month of the gut microbiome, a brain connection, like a whole series of it, and bring on all these people and do an interview and then just drip it out throughout a whole month. It'll be like a 30-day masterclass on the gut
Well, I'm
microbiome.
happy to give you like a whole list more of people, but we are more microbial than we are human, and
it's true, way more.
We need to.
It's like this is just the condominium for our microbes, right? It's like that's what we are. Okay, great. We'll put links for those books you mentioned, and then Martha was on the Keto Camp podcast before, so if you're watching on YouTube, we'll put that video interview so you can watch that next. If you're listening on the podcast, we'll drop that in the notes. We'll put your website and everything you mentioned down below. Last question for you, Martha. My favorite vitamin is vitamin G, gratitude, and I've got a lot of gratitude for you. Your friendship, your support, I'm learning so much from you, and I just want to ask you, what do you got vitamin G for today? What are you grateful for?
Oh, well, I'm truly grateful for you, but I am so grateful for all the people who contact me and like they bring me little nuggets, and they also bring me appreciation. So, I feel wonderful. If there's somebody who has a question and I can help them, I'm grateful for the opportunity to help people. And what I learned from your vitamin G, I sign all my letters to every customer that I write to every person that I, am emailing or whatever, with gratitude, because I am really grateful for the opportunity I have to have this knowledge and to be able to help people feel better.
I love it. Yeah, that's so cool. I love that you do that. There you go, Martha. Thank you so much. Oh, one more thing. Me and Martha are going to be speakers at KetoCon, right? KetoCon is going to be the Super Bowl of keto conferences. It's taking place the third week of April in Austin, Texas. Martha is going to be speaking on, is it molecular mimicry, your topic, or is it something else?
I'll be covering molecular mimicry, but just overall kind of how the microbiome and keto play together, and what these metabolic changes mean for our health in our
awesome.
Yeah.
I can't wait to see your lecture. So Martha's going to be speaking on that. I'm going to be speaking on more of like the cell danger response and ways to support the mitochondria with ancient healing strategies. I'm also doing a meet and greet and a workshop on keto flexing, and I'm going to be hanging out all weekend. So is Martha, and we're doing a Keto Camp meetup. So I'm saying all that because I want to inspire those listening and watching to go get your tickets to KetoCon. We'd love to give you a hug and hang out with you. It's ketocon.org. And I do have a coupon code, which is Keto Camp, for $50 off your ticket price. So go check that out. Martha, I can't wait to have another conversation with you and also see you in KetoCon in a few weeks.
Thank you so much for having me, Ben. It was great to be with you today.
Thank you, Martha. We'll talk soon.